good morning, and good evening, depending where you are. Dear ISV members, global vaccine colleagues, and friends, on behalf of my fellow co-chairs and the entire board of the International Society of Vaccines, I would like to warmly welcome you to today's ISV Virtual Congress on COVID-19 vaccine development. So the next one, I would introduce to you today's chair, Professor Denise Doolan from the James Cook University, Australia. She is also a fellow and board member and officer of ISV. She is also the president-elect for ISV starting next year. So now, let us welcome Denise for her welcome address. Denise, please.
Hello everyone, welcome to today's live broadcast on the 5th COVID vaccine update. It is a great pleasure to welcome you to this virtual congress, which is the 5th in the series of COVID vaccines. So before we start, I just wanted to give a little bit of introduction on the International Society of Vaccines. It is a not-for-profit member organisation spanning the globe. It currently has membership of 750 members from about 39 countries. So ISV's main function is to promote knowledge exchange and build national, regional and international capacity in vaccine science and delivery. And we really encourage everyone who's interested in vaccine science to join the society. Over the last year, the rapid development and rollout of vaccines to combat the global pandemic of COVID-19 has been an amazing scientific achievement. This ISV Virtual Congress Series provides a platform for key COVID-19 vaccine developers to share their progress to the scientific community and global community. Only vaccines that have received regulatory approval for emergency use authorization have been featured in this series of virtual congress. So this Congress today, the 5th in the series, focuses on the Asia Pacific and specifically on the 4 vaccines developed by and approved for use in China. 3 of those vaccines are inactivated vaccines, and 1 is an adenovirus serotype 5 vaccine. So it gives me a great pleasure to now introduce the first speaker, who is Dr. Jinbo from CanSino Biologics. Dr. Go joined CanSino Bio in 2016 as the Director of Clinical Operations, leading the clinical development of the meningitis conjugate vaccine, adenovector Ebola vaccine, the pneumonia polysaccharide conjugate vaccine, the pneumonia protein vaccine, and the Ad5 COVID vaccine. Prior to joining CanSino Bio, Dr. Go was an epidemiologist at Tianjin CDC in China, engaged in infectious disease control and epidemiological research. Today, he will be presenting a talk on the update of the recombinant adeno5 vector COVID-19 vaccine. It's my great pleasure to welcome you to the stage. Thank you.
Okay, thank you, Dr. Lu and Dr. Dennis. Very glad to be here to give you some updated data on the recombinant adenovirus 5 vector-based COVID-19 vaccine manufactured by CanSino Bio. Next slide, please. This is the contents about today's presentation. First, I will give some updates from our current ongoing phase 3 efficacy trial, and this data is from the interim analysis. Next slide, please. And this slide shows the interim analysis data of efficacy, and this data is based on 100 cases, and all these cases are reported at least 28 days after vaccination, and all the cases are confirmed by the independent endpoint review committee. And from the table, you can see in age group 18 years and above, the efficacy against all the symptomatic cases is 65.28, and the lower limit of 95% confidence interval is 45.73, which is higher than the WHO guideline requirement. It is 30% that requirement. In terms of severe cases, the efficacy is more than 90%, and also showed a very significant efficacy against the severe cases. So next slide, please. Okay, this slide also gives the information about the efficacy analysis based on 205 cases. The difference is the old cases is reported at least 14 days after vaccination. And also we give the age group stratified analysis in the table. In the 18 years and above age group, the efficacy against all the symptomatic cases is about 69%, which is much similar as the previous 28 days data. And the efficacy against the severe cases is more than 95%. In age group 80 years old and above, the efficacy is about 68%. And for the severe cases, it's nearly 89%. Because in this database, we have more cases. So you can see the 95% in 95% CI is narrow. And the lower bound of 95% CI is 57.03%, which is much higher than 30%. And for the senior people who is 80 years old and above, it also shows very significant benefits there. Next slide, please. And this slide gives the safety data from the interim analysis in terms of solid-sided AR. By the time of interim analysis, we received nearly 700 diary cards from the extended safety cohort. And from the table, you can see the total solid-sided AR is 69 in the vaccine group and 48.2 in the placebo group. Considering the characteristics of this 85 vector-based platform, we believe that all these adverse reactions, especially the systematic AR, is expected AR, because we have also observed the same, very similar AR in our previous 85 vector Ebola vaccine, which is used the same platform. And all the AR, the average duration of all the systematic AR is about 2 days. And for fever, it's just 1 day. So the AR is transient. The safety is good, relatively good. Next slide, please. And this slide gives the safety data in terms of SAE and MAE. And SAE is serious adverse events, and MAE is medical attended adverse events. So by the time of interim analysis, more than 34,000 subjects were randomized in the trial, and we do not receive the report of related SAE. And you can see in the table, between two groups, the incidence of SAE have no significant difference. For MAE, no matter related or not, the incidence between two groups is also have no significant difference. So next, please. Okay, that's the brief introduction from the interim analysis data of our current phase 3. And next, I will introduce to you the efficacy against the new variants of this vaccine. Next slide, please. We have conducted a cross-neutralizing IC using a human serum immunized with our 85 uncov vaccine. We used the CPE-AC, and 10 various strains are used for testing, including B614G strain, and also include the British strain and South Africa strain, and some other early epidemic strain from China. Next slide, please. And this slide shows the neutralizing data, titer data. You can see very clear here, except one strain, that is South Africa strain. All other 9 variants, their neutralizing antibody titer is almost the same. It seems no significant impact on this, on its neutralizing antibody titer. But for the South Africa strain, the neutralizing antibody titer drops for about 3 or 4 times. So it reminds us that maybe South Africa strain, we need to pay more attention about this strain. Okay, next slide, please. Now I will come to the some discussion of immunity evolution and our plans of booster, something like that. Next slide, please. Children and docents are the next big group for the vaccination all over the world. So we have already started phase 2B clinical trial in China to evaluate the immunogenicity and safety in this age group. In this phase 2B, ongoing phase 2B, we enrolled children and docents from 6 years old to 17 years old. And we also enrolled some senior people who is 56 years old, and we also included those subjects who has been previously vaccinated by the 85 vectored Ebola vaccine. I think the main part of this trial has been finished, and the data has already been generated, and the result expected can be available by the end of April. It is maybe 2 or 3 days later. And we also we are preparing for immunogenicity bridging phase 3 trial in China in children and docent population. I think that big trial will be initiated in China very soon. Okay, next slide. We have also conducted a booster study in the phase 1 subjects in Wuhan, and we gave the booster injection 6 months after the first injection. This slide shows the result in terms of RBD ELISA antibody. You can see this slide, it shows very clear that this vaccine, 85 vector COVID vaccine, can be well boosted after the first dose, from the first dose. It is almost a 10 times increase in antibody titer 14 days after the boost dose compared with the first dose. Okay, next slide, please. So according to the previous data, we have decided to carry out a booster vaccination study in our current phase 3 efficacy trial. And this will also meet the requirement of the local investigator and subjects their requirement. They have the urgent requirement to be vaccinated by the real vaccine, especially for the placebo group. So we we will give all the subjects 1 dose injection of 85 uncold vaccine under the blinded state, and we will follow up them for at least 6 months to observe the relative VE of booster dose against the single dose vaccination. So that will offer us some very valuable data about booster dose of efficacy. And interval between the booster and the first dose is about 6 to 7 months. Okay, next please. Okay, so finally, in this presentation, I will bring some new information about our aerosol vaccine design. Next slide, please. Okay, sometimes we have been asked the question for adenovirus vector-based vaccine, can we do something better than now? And I think one answer to that is we can use the mucosal immunization route instead of intramuscular route. This slide gives the brief and clear introduction on how mucosal immunization works and compared with the intramuscular immunization routes. From this slide, we can see mucosal immunization route can induce strong humoral immunization response, including IgG response and IgA response, and especially those IgA on the mucosal surface. And it can also induce very strong cellular immune response in the lung tissue. We believe this mucosal immunization route may give us an opportunity. That opportunity is to protect from the infection of SARS-CoV-2 instead of just to protect from the COVID disease. It's meaningful for the epidemic control. And also, it has other potential benefits. It eliminates local side reaction, safer and more tolerable. And also, in this mucosal immunization, we just use a little dose, and it will vaccinated in a very fast speed. So that's these advantages. So next, please. Actually, we have conducted a protective efficacy trial in ferrets. This slide shows some results of this animal trial, and from chart A, B, and C, you can see the red dot is the immune response from the mucosal route, immunization route. It is vaccinated first by the initial and then oral immunization route. The blue dot is intramuscular route. From this three chart, we can see no matter humoral immune response or the cellular immune response, the two routes looks very similar. No significant difference between them. But if we see move to chart D and E, which is the challenge trial, there shows some difference between these two routes. The mucosal immunization route shows better protection than the intramuscular route. That is the red line. The red line is mucosal route, and the blue line is intramuscular route. So it's very clear. Next slide, please. We also evaluate the antibody response in NHP. This slide shows the antibody titer after 3 doses of mucosal immunization, both in the initial route or in correlation route. First dose is day 0, second dose is day 14, third dose is at day 28. And you can see the red line in the chart, that is reference data from previous intramuscular antibody level. So after 2 doses, that is antibody level after 2 doses. And we can see 2 dose inhaled administration generates almost the same level of ELISA antibody compared to the 2 dose of intramuscular injection, and even in the very low dosage. In terms of IgG, inhale route is about the same, just as I said, and it's also much more protective than the initial route. So finally, we choose the inhale way as our mucosal immunization route. So next, please. This instrument is what we used in our previous trial. It can generate very small particles, small liquid particles, and can be used for an inhaled clinical trial. Next slide, please. And actually, we have initiated a phase 1 and phase 2 trial in China of inhaled administration route, maybe 10 days ago. This is the brief introduction of this trial, of design of this trial. We use 3 dosage in this trial. That is 0.5 times 10 to the 10th Vp, and 1 times 10 to the 10th Vp, and 2 times 10 to the 10th Vp dosage. We used a 2-dose schedule in this trial, 56 days apart. And we also used a mixed group. That is, first dose, we used the intramuscular injection. And the second dose, we used the inhaled vaccination. And this trial will give us, offer us a lot of information of the inhaled vaccination, and compared to 1 dose intramuscular injection, and gave us the information of mixed group. And we will collect blood sample day 0, day 14, and day 28 after each injection, and 3 months 6 months and 12 months after the second dose. So it will offer us more information about the immunogenicity dynamic data. Next slide, please. Okay, thank you all. That's my last slide. 谢谢大家. Thank you.
Okay. Thank you very much, Dr. Gao. That was a wonderful presentation. So just a few questions that have come through from the question and answer section. One question relates to the blood clots that have been seen with the AstraZeneca vaccine in Europe. The question is whether you see similar adverse events with your vaccine.
You mean the blood clots?
The blood clots, yes.
Blood clots. Yeah, after that news, we also see that news, but we have go through our current clinical trial safety database, and our post-marketing PV team also goes through their safety database. But for now, we did not see the similar cases, either SAE or MAE, something like that. But we keep our eye in this. This is very important. Maybe some very important safety signal. If there is some very important safety signal related to this issue, we will ask the investigator to record it and give us data to analyze. But for now, I think it's okay for now. We did not have received the report about this.
Thank you. And just following up on that, with regard to the inhalation route that you were talking about, do you have safety concerns for the inhalation route of administration?
Actually, we have conducted a lot of preclinical research on the laboratory and also in the animal. For now, we think the safety could be maybe could be better than the intramuscular route because we use relatively low dosage, and it's just a local site immunization response.
Excellent.
Yeah.
Okay.
Maybe it could be better. I think our new phase 1 and phase 2 trial will offer us more information about this.
Thank you. And following up with the mucosal route, is there any evidence about the antibody response? Do you have a reduced antibody response using against the adenovector?
We have not conducted such studies previously, but in our current phase 1, phase 2 trial, we will test the vector antibody level. Yeah.
Okay. Thank you. And another question with regard to boosting. Boosting may interfere with the anti-vector antibodies. How would you propose that problem might be overcome?
I think this is the old question and also been asked to us. There are several ways. When we design the vaccine, we just about the dosage what we used in the final product. So maybe some dosage we can improve some dosage to deal with the pre-existing 85 anti-vector antibody issue, and also something like the mucosal immunization route is another way to overcome this issue. And in our phase 3 trial, efficacy trial, it already been clearly demonstrated that this vaccine can work well in different places over the world, in different places. So I don't think that will be a problem.
Okay. And with regard to the pre-existing immunity, if the anti-vector antibody does limit the boosting, have you considered heterologous prime boost, such as with the recombinant or inactivated vaccine, or possibly a heterologous boost to more easily make boosts for the mutant strains?
Yeah, if we just simply use the 2-dose regimen, for example, first dose day 0 and second dose after 28 days, maybe it's a problem because at that time the anti-vector antibody level is in the very high level. But now we are considering the primary booster schedule, but almost 6 months apart between 2 dosage. And we think the anti-vector antibody level after 6 months is relatively very low and have little impact on the second dose. Yeah.
Have you done extensive investigation of the interval for boosting? Why was 6 months arrived at, and is it possible that longer or shorter intervals may be more efficacious?
Actually, previously there are some studies on this, not only done by us but also by other investigators. It shows that for the adenovirus vector vaccine, it's be better. The second dose be better given to the subject more than 3 months. It will be better. If less than 3 months, maybe it will have some impact on the second dose. Yeah.
Okay.
And yeah.
And just with regard to the dose, do you expect that 1 dose would be sufficient for the inhalation route of administration due to its expected effect on immunogenicity?
For now, I can't give you the very confirmed answer about this question. In the current phase 1, phase 2 trial in China, we used the 2-dose schedule, but we also draw some blood after the 1 dose. So I think those data will offer us the very solid, evidence to tell us how to choose the schedule. Yeah.
Okay. And a question with regard to the CD8 T-cell responses to your vaccine, and in particular, how they respond to the variants. Do you have any evidence, and in particular with regard to your data on the South African strain?
I have one slide about the cross-neutralization AC we conducted in China. There are 10 strains. One of them is South African strain. We can see some, antibody drops against the South African strain. But I think this is a common, problem across maybe all the vaccines. So we are preparing. Internally we already preparing for the updated new generation, updated the vaccine to cover this problem.
Okay. And the last question, can you comment on the immunogenicity observed in your phase 1, phase 2 studies in China compared to the observed responses overseas in phase 3?
For now, I can't give the answer because for now the serum from the phase 3 is in the laboratory, central laboratory in Canada. It's in progress testing. So we did not have that data yet. So after we got the data, I think we will compare with the phase 1, phase 2 trial data. But I think it's expected it will be very similar because we did not change our vaccine in phase 3. Yeah.
Excellent. Well, thank you very much for a very stimulating talk, Dr. Go. I will now hand over to Dr. Shan. Thank you very much.
Thank you.
Thank you, Dr. Go, for a very interesting talk. So now we not only learned what you have done with your vaccine with the phase 3 trial, but also we are learning the interesting mucosal delivery.